Okay, so part of the exam spec is to look at microscopes. Now, of course, you can't do that at home whilst we're on lockdown. So I'll try to cover as much as I can in this video. Now, microscopes really have three main parts. You've got the eyepiece lens, which is the bit we're looking down. You've got the objective lens, which we can rotate, and we can select different levels of magnification. So say times 4, times 10, times 20. And then we've got the stage with the light, and this is where the sample we're trying to view would actually go. So eyepiece lens, objective lens, the stage with a light source. Now, if I wanted to work out the total magnification for this particular microscope, okay, if we look, if I was using this lens, the times 20, and I know my eyepiece lens is times 10, all you do is you do 20 times the 10. So 20 times 10. So that is times 200. Excuse me. So the total magnification for that particular microscope is times 200. So that will make an object appear 200 times bigger. It won't make it 200 times bigger, but it will make it appear 200 times bigger. So remember, we're using microscopes to look at microscopic things, very small things. It comes from a Greek word, micros, which means small. Now, another thing they may ask you to do is to identify some cells that have been, pictures maybe that have been taken from under a microscope. So the typical one we normally do in class is to extract some of your cheek cells and have a look at some onion cells as well. Obviously, we can't do that from home. The typical cheek cell just looks like a standard animal cell. So the main thing is maybe identify some of the structures. So we've got the cell membrane, we've got the nucleus, and we've got the cytoplasm. Onion cells, obviously onions grow below the ground, so don't expect to see any green chloroplasts. So you typically tend to see the cell wall and generally see the vacuole, and occasionally you may see the odd nucleus. It depends on the quality of the sample. The cell membrane is normally tucked just on the inside of the cell wall there. So we'll see the cell wall, the cell membrane, the nucleus, and the vacuole. Definitely not chloroplasts. So don't try to fake it and try to identify chloroplasts. This is an onion, it grows below ground, no sunlight, no need for chloroplasts. Okay, now sometimes there's a calculation involved where they'll tell you two things. They'll tell you normally the size of an image, so maybe the width of this particular cell, or they might describe the diameter of the nucleus, and they'll probably tell you the actual size of a nucleus in real life, or an actual length of an onion cell in real life. And they normally ask you to calculate magnification. So what we do is we use this formula triangle, I am, nice and easy to remember, I am. So if we want to work out, say, A, it's I divided by M. If we want to work out M, it's I divided by A. And if we want to work out I, it's A times M. Now, because they normally want us to work out M, it's dead easy. It's I divided by A. So I is the image. It's normally the image in the exam paper or on the screen of the cell. A is the actual. So that's the kind of the real life size. And M is the magnification. Okay. So if you were told, for example, that the image size, I don't know, was 10 and the actual was 1, then to work out the M, you do 10 divided by the 1. Okay, so I hope that helps in terms of microscope work. The main parts of the microscope, how we calculate the total magnification, how we can calculate magnification, and then maybe identifying cheek cells and also some onion cells. Thank you.